Guess I'll go ahead and get started. Welcome, everyone, and thanks for joining us for our Research 101 webinar. I'm Annamaria Scaccia, board member at COA. Joining us are Katie Coleman, the founder of COA, and Dr. Elizabeth Lisa Henske, who will take us behind the scenes of how a research lab works. This session is being recorded, and we'll have a Q&A at the end, so drop your questions using the Q&A feature or chat at any time. But actually, please leave the chat for conversation. I misspoke there. Q&A for questions, chat for conversation. And that is the last that I will say to it about that. I'm going to go off camera and on mute, and I'm going to turn it over to you, Katie.
Yeah, thanks, Annamaria, for kicking us off. And Dr. Henske, thanks for joining us. We're really excited about this webinar and to learn a little bit about what goes on behind the scenes of a lab and everything that goes into the work that eventually ends up coming out of your lab and in all the papers that we see. So to kick us off, can you briefly introduce yourself and share with us where your lab is located?
Yes, thank you, Katie and Annamaria. I'm super happy to be here. I've never done anything quite like this, so I'm really looking forward to it. So my name is Elizabeth Henske, but I've always gone by Lisa, and I am a professor of medicine at Harvard Medical School and a medical oncologist. I see individuals with kidney cancer and prostate cancer at the Dana-Farber Cancer Institute. And most of my time is spent on research in a laboratory. And my laboratory is at the Brigham and Women's Hospital, which is in Boston, right next to Harvard Medical School. And I'm in my lab right now.
And I think one of the most exciting parts of having you join us is the fact that I think your lab is the largest, if not the largest, probably studying chromophobe kidney cancer. So it's really exciting. One, to have seen all the work that's come out of your lab. I think has there been 4 papers this year that has come out of your lab, or 3 papers that have come out? So we're really excited to see the progress moving. And I was wondering, as we start diving in here, if you might be able to kick us off by sharing a little bit about the basics behind what a research lab does every day. What does the day-to-day look like inside your lab?
Well, I can tell you about today as maybe a kind of typical example of what a day is like. So today I met individually one-on-one with 2 different lab members. One of them is working on a project that's still very much underway, looking for proteins in the blood that might be markers, biomarkers, we call them, of chromophobe kidney cancer. So we were talking about the data and how to maybe present that in a paper, and we've just recently submitted what's called an abstract, a very short 1-page summary of this work. And after that, I met with another lab member, and in between had briefer conversations with almost everybody in the lab. The lab has maybe a dozen or so people. And then just 2 hours ago, we had our weekly lab meeting. So a lab meeting is kind of a big deal for a lab. Everybody gets together, and the way we run our lab meeting is that 2 people each present some of their most recent data, each for about half an hour. So both of those presentations were related to chromophobe kidney cancer. And then we do something called a journal club. So a journal club is when we look very carefully at a paper published by another group. And so today that paper happened to be about ferroptosis, which is something we're very interested in terms of chromophobe kidney cancer. So the lab meeting is kind of the high point of the week because everyone gets together. I always bring snacks for the lab meeting, and it's a chance for everyone to join together talking about the most recent data and science, and it's fun.
Yeah, that's exciting. So it sounds like you kind of have these meetings to get caught up to speed and kind of progress the ideas as the different experiments go along. Was I following that kind of correctly as far as your normal cadence? And then once you get to a certain point, you're submitting the abstracts. And We've highlighted through COA's previous videos doing our research in motion, where we highlight the posters that are at different medical conferences like ASCO GU and various places. When you submit the abstracts, are you submitting those for papers that eventually will come out of it, or are you submitting them for kind of these posters that get presented at conferences?
Yeah, you're exactly right. So the first step is often to present the work as a poster at a conference. And that's a great chance for the person doing the project to get feedback from other colleagues and other scientists. And then ideally, that will lead to feedback that will help to improve the work or add in new ideas. And then eventually that will lead to a manuscript, which is a publication that gets submitted to a research journal and then reviewed by usually 3 other scientists to get feedback, and that will lead to ultimately to a paper.
Yeah. It's always exciting to me. I didn't know anything about this space beforehand, so it's always exciting for me to learn all the nuts and bolts behind all the steps along the process. So one of the things that I know when I initially started learning more about this space that I didn't know was the difference between basic translational and clinical research. Can you share with us a little bit about what these different kinds of research are, and then what your lab primarily focuses on?
Yes, I think when I think about, people use these terms somewhat differently, basic, translational, and clinical. In my mind, basic research in chromophobe kidney cancer is to understand what is causing this particular type of kidney cancer that seems so different from any other type of kidney cancer, for that matter, any other kind of cancer at all. And then translational, I think about things that we can do in cells or sometimes in animal models like mice. What can we do to understand how to take what we've learned about the cause of chromophobe kidney cancer, for example, and then use that to test different possible treatments? And those 2 things are what my lab is focused on. And then the third, of course, really important step is to test these ideas in people in the context of a clinical trial, to see if what we can learn from studying cells and mice might be relevant to actually helping individuals who have this type of cancer.
Yeah, so I know earlier you mentioned kind of some work exploring biomarkers, for example. And with the work that you're doing kind of in the lab, that one would fall into kind of that translational category where you're using mechanisms and understanding how the biology works to trying to compare that with tracking clinical outcomes, but not quite in a clinical trial yet, to the point that you're tracking that directly kind of in along with treatments for patients. Is that where that one would fall?
Exactly. I would put that one in the translational bucket.
Awesome. So you highlighted a couple of the people in your lab that you kind of worked with today. Who are the kind of people that work in the lab? What are the different roles, and what do each of them do? Kind of, I know there's postdocs that are in a lab. There's physician scientists. I know there's PhD students. So can you talk a little bit about the different roles that it takes to kind of have a full lab?
Yeah, there's a lot of different types of people, at least in our lab here. So that runs from people who graduated from college just a year or so ago. We have 2 people in that sort of category who are planning to go on to usually to get a PhD later on. So it runs from those people to very established PhD scientists who have their own faculty appointments, but are still sort of part of our group. So we have 2 people in my group in that category, and then actually 3. And then we have in between people who have PhDs or MDs who are generally here for a shorter time. And some of them are individuals with MDs who are doing research before going on to their residencies and their like internal medicine residencies who actually hope to be oncology scientists, oncology physician scientists. So this is an opportunity for them to have a really intense research experience, learn how cancer research works, and then bring that forward as they go on in their own careers.
Yeah, I know when I visited your lab, I spoke with a few individuals that were kind of in that category that were going to go on to residency, and I believe are in their residency now as well too. And I found it really inspiring to not only see the energy about the passion for the work that they had done over the past few years, but also knowing that they're going to take that into their training as they continue on. I mean, I've not a doctor myself, but I can only imagine that like having that understanding and that base framework really helps them as they progress into residency and eventually to become oncologists as well too for a really fully rounded picture. So speaking of kind of the progression from all of the different folks that it takes in the lab, can you share a little bit about what it takes to bring a project to life from the idea, kind of conception, to a published paper? And then also talk about kind of that process can take a bit of time. So speaking to what are the pieces that take time in between. And I know you had a few slides. I don't know if this might be a good place to show some of those too.
Yeah, I'd love to. Let me just say that it is a very, very long path in most cases, like years between an initial idea and actually identifying. Oh, no problem. Between an initial idea and having something that really has come forward to be published. And that's because the only things we want to have published are the things that we're really, really confident about. And many really great ideas don't pan out for one reason or another. And that could be because the hypothesis was wrong or because we don't have the right tools and reagents to sort of make it work at that particular moment. But let me show you just an example of what an experiment looks like. Well, first of all, you asked about the people in the group. Are you seeing my screen now, Katie? So let me show you this is a picture of our group at the Museum of Fine Arts in Boston. If you've ever been there, you might have seen this amazing statue in the middle. But this includes people from all of those categories, people who are established, what we call junior faculty members, people who have faculty appointments but are earlier in their career, all the way to people who have recently finished college. And then, a key part and a wonderful, wonderful part of what we do in chromophobe and in many other situations is just working with really committed, smart, wonderful people from all over the world. And in terms of chromophobe kidney cancer, that includes people who you may know, Katie, and other people on the line may know David Braun, who's now at Yale, Vincent Zhu. He's the one working on the biomarkers with us. He's at the Dana-Farber. Tony Shaweri, who's one of the most generous, wonderful oncologists I've ever met. Bill Sellers, who's helping us also on a chromophobe project. So just lots of great people are involved, and people are just really generous and wonderful. So let me show you an example of an experiment. Oh, I just would say, like, you might be wondering, why do we care so much why chromophobe kidney cancer occurs? Like, why don't we just find a treatment? Well, we think that really one of the best ways to find a treatment is to understand why a tumor develops. And this, it's not just that I think this, it's that this is really how it happens. So for example, the 2018 Nobel Prize was given for the discovery of how immune cells are involved in cancer. This was something that emerged over 20 years or so of research to understand what's happening with T cells, for example. And of course, that's the foundation for the immune therapy that now benefits many, many individuals living with cancer. And then the Nobel Prize in 2020 was given to, actually, I can't see what year it was on my slide right now, but 2019 rather was given for to Bill Kalin, who's here at the Dana-Farber, and others, for understanding how tumor cells respond to oxygen, and that led to the identification of treatments for kidney cancer, the so-called tyrosine kinase inhibitors. So really important things can happen in terms of basic research leading to translational research, leading to clinical trials that demonstrate true benefit. So I only have 3 slides. This is the last one. I want to show you an experiment that Samer Salem did that are in a paper that he published about 6 or 9 months ago. Samer looks very, very serious in this picture because he has now left my lab and he's now an intern at the Cleveland Clinic, and this is his intern photograph. But he's also a really fun. He's not always this serious. So anyhow, this experiment is so complicated. So he was trying to understand something about the links between ferroptosis and a particular mechanism that he discovered in the lab might be important. And that mechanism involves a protein called ferroptosis suppressor protein 1. So he did this experiment, and he did it in triplicate, meaning that every single part of it was done 3 times. And so what you're seeing here, each of these little boxes is basically one experiment. And this experiment was done in a plate that allowed him to run 96 things at the same time. So first he had to put the exact same number of cells in each of these little, what we call them a well. This whole thing is about the size of your palm. So he had to meticulously put the exact same number of cells in every single of these wells. These are 2 different types of chromophobe kidney cancer cell lines. And he compared that to a clear cell kidney cancer cell line. So first he puts all these cells in, then he lets the cells grow overnight in a specialized incubator that has just the right amount of carbon dioxide and oxygen to let the cells grow. And then he had to add to each of these either this drug, which inhibits FSP1, it's called FSEN1, at all these different doses. These are 10 different doses, or this drug, RSL3, at all these different doses. And then the next day, measure whether the cells were alive or not. And so if it's a purple color, then that means that these particular cells did not survive the combination of this drug plus this drug. So he did this in triplicate, meaning he did this 3 separate times with these cells, 3 separate times for these cells, and 3 separate times for these cells. And out of all this, he learned that if you combine these 2 drugs, RSL3 and FSEN1, you can decrease the survival of the chromophobe kidney cancer cells. So the important thing here is not the result, it's really the meticulous way that he combined all these different doses of the 2 drugs to really understand that these 2 drugs could work together. And this is just one out of many, many experiments in his paper. And even this experiment, it took a lot of work to get to the point where he even realized that it might make sense to combine these 2 drugs. So this is just one tiny part of a much bigger story, kind of, than what's reflected here in this one experiment. So I think just to answer your question, experiments take a lot of time, and that time is not only the hands-on time, it's the thought that goes into it. It's all the things that don't work, that you try, that you don't ever even make it into the paper because something went wrong, or it wasn't as you expected it to be, or the hypothesis was wrong. That happens all the time also. In this case, the hypothesis was right and the experiments came together, and we think it is a really important step for future treatment of chromophobe kidney cancer.
Yeah, I think that's really exciting to see the progression of how that happens, especially because following the research that's come out of your lab for a number of years, I know some of the initial work on ferroptosis talked about some of the initial agents that induce ferroptosis, I believe like IKE or Some of the ones that are more directly targeting some of the pathways that had been previously studied. And we recently released a video on COA's YouTube channel highlighting this paper and talking about how that led to higher toxicities. So that's basic science currently, so not in patients. But if that were in a clinic, those treatments likely would be too toxic to patients. And so if you go back to the lab, and it sounds like Sommer's idea here is, can we combine multiple agents possibly and where with lower doses and see what the effect was? And so I really found that neat as I was both speaking to him and now seeing you present
Mhm.
the research and the charts there as well too, is helping to identify those things early on so that you can understand that by understanding how the biology works and how you can make sure you're kind of using the lowest doses that you possibly can to create the desired effect before you start moving along the line into translational and clinical research that goes on to develop treatments. So my next question is kind of furthering going down the line on that continuum. So let's use his paper as an example. What are the next steps that usually follow that kind of research being published? Is that where, because I don't believe these agents are currently available in clinical trials yet. And so what is the process to get from that kind of more basic and translational research into clinical trials? Is that something that you watch to see if new agents come online for clinical trials for other disease types, or what does the progression process usually look like from there?
Yeah, I mean, that's really at the heart of where we are right now. So, as you've already mentioned, and I mentioned also, we're very interested in the possibility that ferroptosis could be a good pathway to the treatment of chromophobe kidney cancer. But a huge but is that we don't have a drug right now. And other people have tried very hard to adapt the drugs that we've been using in the lab to make them amenable and have a longer half-life, for example, and be tolerable. And it just seems like they're not going to be able to do that. So this new drug, the one that was used in that slide from Sommer that I showed, that seems to be a direction that the ferroptosis field is moving for agents, because ferroptosis is not just important in chromophobe kidney cancer, it's important in many other types of cancer, which is a good thing because pharmaceutical companies may move something forward for more common diseases, and then that could benefit the chromophobe community. But I think it's, if we have to develop a completely new drug, that's a relatively long path ordinarily. So as you mentioned, we're always keeping our eye open for maybe a drug that was approved already or is close to approval for something else might be the right drug for chromophobe kidney cancer. So we want both things. We want to be, you know, looking out for anything that's already developed, and then also working hard to develop drugs that might be very, very useful in chromophobe kidney cancer. And one way that I'm trying to help make that happen is by being connected to the ferroptosis scientific community. We want to make sure that they know about chromophobe kidney cancer, and they do now. I think that we've been successful in having them be aware of chromophobe and chromophobe kidney cancer, and as they're developing these drugs, to be thinking about clinical trials that would include individuals with chromophobe kidney cancer. But those drugs haven't made it to clinical trials yet for any disease. So we want to be on the lookout for anything that might fall into our lap that would be a good treatment.
Yeah, I think there was a couple things that came to mind as I was hearing you talk about that. One was, at the beginning, you were telling us how one of your how you do the journal clubs on a paper and how that was
Mhm.
on ferroptosis this time. So
Mhm.
that's kind of that in action of kind of keeping an eye out, it sounds like, for what other groups are publishing on ferroptosis to make sure that you're can either take that back in the lab or understand kind of where that's following with this wider group that's studying ferroptosis as well too. And then the other piece that it made me think of when you mentioned the benefit of being able to publish this work so other groups who may not be working on even kidney cancer can be aware that chromophobe is sensitive to that. I think of something that I ran into earlier this year was when I was at the AACR annual meeting, which is the big kind of scientific conference. And it was very shortly after your paper the paper had just been published on targeting c-Kit. And one of the Scholar in Training Awards that was awarded for chromophobe at that conference was on targeting c-Kit, that research that came out of the lab. And I had somebody who came and found me because they know that I'm the person that talks about chromophobe all the time. And somebody happened to mention that there was a trial targeting c-Kit. And this was a phase 1 trial, so it does include a bunch of other types of cancer, but chromophobe was listed as one of the designations of that. And so it's, I think, really neat to hear how important it is and how connected the space is. And how important it is to share the research. So even if there's not new treatments that are available yet, that was kind of serendipity that both of those things happened at the same time, that there was this trial that was opening, targeting a very similar pathway to what was being explored in the research. I don't think it always happens like that, but publishing on it and making sure these communities are all aware raises the chances of those kind of overlaps happening. So I thought that was a really neat thing that I was able to see kind of play out earlier this year that really speaks to exactly what you were highlighting there.
I think I think visibility of any rare tumor type is so important. So it also illustrates how you're raising awareness about chromophobe, and other individuals and foundations also can really move us forward. So, and that if we're all talking about chromophobe, which I do every place I go, I'm talking about chromophobe kidney cancer, it increases the chance that we'll have, two things come together that wouldn't have come together otherwise. I also would say that chromophobe kidney cancer is so unusual and distinctive that any scientist you mention it to and start to talk about the features of it, they're so curious. They which is a really good thing. So we want many people to be interested in what's the cause of chromophobe kidney cancer, why does it happen, and how can we effectively treat it. And the more you talk about it, the more people are interested and might recognize that some part of what they do could be relevant.
Yeah, I think that's really what's exciting as far as kind of these webinars, the work that you've done, how much this community has grown over the years. Even everybody on this webinar attending, that actually really raises visibility. The fact that there are people interested in hearing what the latest research is and how this happens, all of that helps. Literally, even the registrations help, I'll say, as running COA, being able to share with people that people attend our webinars and it's a topic that people are interested in, makes it so that we get more support and more people wanting to join the webinars and help share the content as well too. So really takes all hands. I'm very grateful for the work that your lab has done to really kind of provide more structure to the basic science of chromophobe, and really has kind of pushed everything along in the past few years that all of our efforts have really been able to help rally around as well too. So one of the things that you talked about a little bit as well too is the importance of that when it comes to funding this research. So we know chromophobe is kind of unique and very different from other types of kidney cancer, for example, for which most of the treatments are currently derived. Can you talk a little bit about who funds the research, both research that's done in a lab like yours, as well as what funding kind of goes into bringing new treatments to patients, either through clinical trials testing existing drugs or creating new treatments as a whole as well?
Yeah, so funding a lab, funding research on anything, and funding research on specifically on chromophobe is that's what I think about all the time, because without funding, there is no lab, but there's no research. So, how do we do it? So I can tell you how we do it today, and I'm not sure how we will be doing it 3 years from now. Right now, about half of our funding comes from the federal government, and half of it comes from donors and or foundations. And the half that comes from the federal government comes from the Department of Defense Kidney Cancer Research Program, the KCRP, and they are supporting research at several different levels, including the development of therapeutic antibodies targeting KIT. So that has really, that program has really helped sort of put chromophobe kidney cancer on the map. It gives us, but anyone else who has a good idea, a chance to move that idea forward. And the support from foundations has been really important because that allows us to get the first, and from donors, to get the first evidence that we're on a good path. And that makes us competitive for grants, for example, from the Kidney Cancer Research Program. So I often think about the fact that an investment of, say, $50,000 can give us the data that we need to get $1 million federal grant to study chromophobe kidney cancer. So that's how things are today, and that's how we've made the progress that's led us to 2025. The problem is that the Kidney Cancer Research Program was, as you know, and I'm sure everyone on the call knows, was completely eliminated. So we don't have any of that type of funding moving forward. We still have the National Institutes of Health, but it's really challenging to get research on something like chromophobe kidney cancer supported by the NIH. We have been successful only once, and we haven't had success since then. So I think it'll be very, very interesting to see how do we support research on the less common types of kidney cancer moving forward. And it's, I'm sure something will be figured out, but I don't know as of today what that might be.
Yeah, and I think it's, I appreciate you breaking down what the different types of funding are. I know so when I first started advocating for chromophobe kidney cancer, and there was a group of us that kind of helped fund the first foundation-led grant for chromophobe kidney cancer, and that one was $50,000 through Casey Cure. That when I initially got involved in fundraising for those efforts, I didn't know all of the behind the scenes of kind of what happens as far as cost-wise in a lab. So that it sounds like that kind of funding is really helpful in being able to run the experiments and being able to get that initial data, kind of like the paper that we saw that was testing the different doses of treatments. But the larger government grants are really what kind of pay for the people in the lab and the lab space to be able to do the work. Otherwise, we wouldn't have because $50,000 isn't enough to pay for a PhD student or a postdoc in a lab in order to carry out those projects. Am I kind of understanding the full kind of picture there in the landscape of funding?
That's right. So a $50,000 award from a foundation is huge, of course, and really can make a big difference. But it's not enough to bring to completion a project, because a project takes multiple people. We need to cover their salaries and multiple reagents, and so yes, it is. It's a big undertaking to make progress.
I think it kind of just highlights even more of how important it is to make this um, a group effort, right? Like it takes everybody to kind of move these things forward and takes all players. I know there's other foundations as well that do a lot of advocating for federal funding as well too. So if there's anybody on the call here, I'm happy to connect with those other groups as well for anybody that's interested in those areas. I I think that's one of the things that makes me excited is that I have seen the all hands on deck approach with chromophobe, and it really makes me excited about um, the progress to be able to continue making advancements for it. So that kind of highlights the funding for a lab. What about the funding for, so once we get past the basic science and translational science and more into clinical research, what kind of funding is typically needed to bring clinical trials to life? And who is typically providing the funding for those trials?
So once things get to the clinical trial stage, sometimes those studies are funded by the company that has the particular drug that's being tested. There also are situations where a clinical trial can be supported by the federal government, and that can be through a grant to an individual who was planning to run the trial, or sometimes through what are referred to as cooperative groups that are involved in running many trials for different cancers. So I think that all depends on what the drug is and what exactly the goal of the study is. For example, the trial using the c-Kit antibody drug conjugates that you mentioned earlier, Katie, is supported by the company, which happens to be located in Korea, that has made that drug and wants to understand if it's working. But it really depends on the exact circumstances. And of course, we've never had a trial that was only for individuals with chromophobe kidney cancer. It's always been chromophobe kidney cancer has always been included with other types of kidney cancer. So I think that's another important step that we may need to take at some point. We may realize that chromophobe is so different than other types of kidney cancer that we really need a trial that would only include individuals with chromophobe. And I don't know right now how that would be supported. I think that would be in some ways a good problem to have if we knew that we had a drug that really would be a breakthrough for chromophobe kidney cancer. That would be a wonderful situation to be in, and I'm sure we would figure out how to get that trial done.
Yeah. So I think that kind of leads into one of the next questions I had, which is I was wondering if you could share a little bit about what makes studying rare cancers like chromophobe kidney cancer kind of uniquely difficult, both in terms of what kind of difficulties do these rare cancers face when studying them in a lab, and then kind of as we were just speaking to the trials and getting the right enough number of patients to be able to enroll in those trials as well too.
Mm. So let me just tell you how I got started. I became interested in chromophobe kidney cancer. It's because I'm interested in two other rare diseases that can be associated with chromophobe kidney cancer. One of those is called tuberous sclerosis complex, and the other is called Birt-Hogg-Debye syndrome, or BHD. So in all of these situations of rare diseases, one of the challenges is always having the right tools. And those tools that we need include cells. We call them cell lines. Those are cells that we can grow in the lab, like those ones that I showed you on Sommer's experiment. So we need cells, and we really like to have mice that have a disease that resembles the disease in humans, so that when we have a good idea, we're not just checking it in cells, we can actually test it in a mouse. So we're really limited for chromophobe kidney cancer in both of those situations. So we have some cells, you saw a couple of them, but we're actually not sure whether those are representative of chromophobe kidney cancer in general. Those are only 2 cell lines. They've been grown, each of them, for maybe 10 years. And each time we grow them in the lab, they're probably changing a little bit. And we don't have a mouse model that we think is a perfect model of chromophobe kidney cancer. So the way that I tend to sort of get past that is we test some things in cells, and then we go back to the human tumors and see, does it match? Does it make sense? And we have a couple of very strong resources in chromophobe kidney cancer. One of them is the Cancer Genome Atlas, or the TCGA, which thanks to efforts by Kim Rathmel and others, Marston Linehan, includes chromophobe kidney cancer. We're so lucky that chromophobe kidney cancer is included in this very important database of human cancer. So we can go back there and say, well, does this idea make sense in terms of what we're seeing, what genes we see are up or down regulated in chromophobe kidney cancer in that data set? And then we also have data from the metabolism of kidney cancer, of chromophobe kidney cancer, which is how we got started on the ferroptosis idea. So I think a limitation is the tools that we have, but we can't just twiddle our thumbs and wait for better tools to become available. So we're always trying to push forward with what we have and make these continuous connections between what we're doing in the lab and what we can see happening in the tumor specimens that we have available to us.
So I think you highlighted a little bit about the models that are used. And I know every time we do a webinar, either through COA or through other groups, I often see patients asking how they can get involved. What are the most useful ways that patients can get involved to help move these kinds of efforts forward?
Well, one important way is just to be aware of what's happening in research. So for everyone who's on the line today, that's fantastic. Another way is to support medical research that's at the federal level through the NIH and the Department of Defense. And then actually I've been exchanging emails even today with an individual who has chromophobe kidney cancer, who has the possibility of letting us access tissue from both the primary tumor and also a site where the tumor had metastasized. So as you may know, when you have a tumor removed, the tissue is stored in the pathology department at least for 7 years, but at a teaching hospital or an academic medical center, stored for, like at the Mayo Clinic, they've been storing things for 100 years or something. So that tissue exists. And one thing that's a need right now for understanding chromophobe is what's the difference between the tumor when it's in the kidney and that same tumor when it has spread to the lung or the liver. It's surprisingly difficult to get those matched samples. So from the same individual, the initial tumor and removal of a metastasis. So that's an example where an individual, we've looked at our hospital and we could only find a handful of those matched specimens because often someone had the primary tumor removed at another hospital, not here. So I think people who could help us find those sort of matches is one way that we could help move things forward. But the most important thing is just to be aware of the research and supporting it in any way that you can, because I think we really recognize that research is the way to treat and ultimately cure cancer.
Yeah, I think it's exciting to know that I know as a patient myself, when I was having my tumors removed, I remember how important it was for me to have my tumors studied. I think it's a desire that a lot of patients have, but they just don't know how to get involved in it. So anyone on the call can feel free to reach out to COA as well too. We're working on programs to be able to help put together which labs can accept tissue and what kinds of tissue, and how to coordinate those efforts as well too. So feel free if anybody wants to kind of reach out and be provided with any resources. There's some paths that already exist for donating fresh tissue. For example, pattern.org allows patients to donate tissue directly from a surgery, but we're always looking for new ways to be able to help connect all of these labs and patients who want to donate for the ones that do. So I think that's really exciting. And then also couldn't agree more on just the fact that being aware of what's being done. I know myself, I did not understand this world at all before I got involved in it and have become quite passionate about it now. But that's because of how important it is. And it's so exciting to see the progress and see things moving forward. I remember when I first started advocating for chromophobe, hearing how long it took to see any progress. And the fact that it's been 5 years since I really kind of started advocating, but there is real progress. And that's really exciting and it's really motivating for me to see. And the momentum just seems like it's picking up more and more every year. So thank you for everything you've done to help move that forward. So as we wrap up, before we move on to a few questions here, are there any takeaways that you'd like the audience to remember, or any words you'd like to share with any of the patients on the call?
I would just echo what you said, that we really are making progress. And one small way to sort of metric that is the meeting that Katie and I were just at, called the International Kidney Cancer Symposium, IKCS, where this year there were multiple talks about chromophobe and multiple poster presentations about chromophobe. So, and that was not the case just 4 years ago. So we're recognized now, chromophobe research is recognized as a very important part, and people are paying attention and asking questions and really helping us move things forward. I know that you sometimes use the phrase "it takes a village," and I use that phrase all the time. We want there to be lots and lots and lots of smart people thinking about chromophobe. We don't know where the breakthrough idea will come from, but the more people who are aware of it and thinking about it, the better. And we are definitely making progress in building that village.
Yeah, super exciting. So I think we'll invite Annamaria back to help us go through some of the questions that have rolled in. So I'll turn it back over to you, Annamaria.
Sure thing. Thank you so much for that riveting conversation. I know I learned a lot. I'm still learning a lot about research and the whole process and everything. So thank you so much. Well, this is our Q&A section. We actually had a few come in, so I'll start with the earliest that came in and work my way down. The first one is, do you have anybody studying specifically sarcomatoid features in chromophobe?
Yes, we do. Actually, right before this meeting, someone came in to ask if I had time to talk about what she's doing with our sarcomatoid project. And I said, oh, we'll have to do it tomorrow. So now it's on my schedule for 3 o'clock tomorrow. But what we're doing is comparing the genes that are expressed by sarcomatoid and chromophobe side by side. And we're very excited about this. We're using the latest technology that allows us to look at individual cells and see all the RNA molecules that they're making. So yes, we are working on that, and other groups, I'm sure, are working on that also. It's a really important aspect of chromophobe biology that seems to predict a more rapid growth behavior.
Well, that's fantastic that you have people studying that, because I know it's something that in our conversations at COA often comes up and we're often asked about from patients. Now, this one's for either one of you, actually. Are there any biology or oncology resources, books or otherwise, that you recommend for patients keen to have a better understanding of their diseases? And ultimately, like understanding current research and scientific papers, not just the abstracts.
I'll let Dr. Henske start with that one.
Well, you could read Katie's book, which I loved, but I guess that's not going to explain the science necessarily. I think it's hard. It's hard for me often to understand complicated scientific concepts, and I often have to hear them multiple times before I finally understand it. So I don't know if there's any shortcuts to that, Katie, that you can suggest.
Yeah, I would say as somebody who didn't have a background in this at all, and who it has been very difficult for me to learn as well too. Repetition is pretty much how I've learned. And so kind of watching some of the videos multiple times, reading the papers, oftentimes I will read a paper and then like now we've got the videos on through Chromophobe, the COA's YouTube channel that will recap these papers, talking with the researchers who are doing the work, and then go back and read the paper again. And I know for me, for example, there was even something just this week where I was reading a different paper that was talking about a concept that got brought up in one of the other ones. And so when those moments happen for me, I try to capitalize on them because it ends up being like a spark of interest. And I have to say, oh, wait, I've seen that before. And then I'll go relearn again for a second. So I'd say we're trying to make the videos on COA's YouTube channel a little bit more approachable, where we're still talking about the scientific findings from a scientific lens and aspect, but also trying to break them down in analogies that might be slightly easier to understand. So that might be one place. But yeah, I think for me, the biggest thing has just been kind of reading it multiple times, kind of watching any videos. I'm a very visual learner, so I love to watch videos and listen to content on things. And then always feel free to reach out to me. I can't advise on anything from a individual perspective, but happy to share anything that I've learned along the way. And in case I might be able to share it in a way that can break it down to the fundamentals, since I really had to learn piecemeal along the way, and I've kind of just had to learn bit by bit too. But I think it's great that for anybody to be interested in this area.
Sure. And I just want to add, there are also advocacy organizations that do, in addition to like COA, right, that do write about studies, and they write it in a way that is more accessible to a broader audience and to survivors and patients. So, oh, not that the question was posed to me, but this is also something I do in my day job. I would say check like advocacy organizations for their blogs, because they often break down these studies too and what they mean in an accessible way. And if you are willing to reach out to the researcher, one thing, I work with researchers all day, but in leadership consulting, and usually when I have to translate their research, I always tell them to explain it to me like I'm a 5-year-old. So if you feel comfortable saying that to a researcher of a study, I suggest also doing that. But I know we don't have a lot of time, so I will move on. What do you interpret as indicators of recurrence for chromophobe following a radical nephrectomy?
Yeah, so we know a bit about factors that are in the tumor, the original tumor, that do seem to make recurrence more likely, but that's in the context of most chromophobe kidney cancers not recurring. So it's important to keep that in mind. But the sarcomatoid morphology, for example, the sarcomatoid pattern that was mentioned earlier, is one factor that seems to make the possibility of recurrence more likely. And mutations in a particular gene, which is called the P53 gene, also seems to increase the likelihood of the disease recurring or metastasizing. But again, we know for sure that 95% of chromophobe kidney cancer will not recur.
Well, thank you for that. I wanted to actually go back to the tissue comment, because that was like last. So one of our lovely participants is in Australia, and they wanted to know if samples from other countries can be of any help. Just to give a little background, because she gave a little background, she has 2 samples at the same hospital. They believe her spread through seeding, and she'll have another surgery early next year. So would samples from other countries be of any help?
Yeah, it's tricky. There are a lot of barriers to collecting tissue from other hospitals, and I'm wondering if those barriers might be even more challenging for tissue from another country. That's why when we wanted to get these matched pairs of the kidney tumor and a metastasis, we started with our own hospital because then we don't have to go through as many hurdles. So I don't know. I think it might be tricky, but we never know until we try, I guess.
And this is actually a follow-up to it from someone in France because they found programs where you could donate tissue, but it's unfortunately just limited to people in the US, which is honestly a shame that it gets so complicated because imagine how much we can do. Do you know if that's something that could change in the not so distant future?
Yeah, I think maybe Katie, you would know if there are any initiatives like that.
Yeah, I haven't found any international ones yet. It is tough. It is a lot easier to do here in the States, with the labs that are working with it in the States. But that is one of the initiatives that COA is working on, is to put together a list of anybody that can accept tissue, where they can accept it from, not just in the States, but all around the world. And so if we can find labs that are interested in accepting tissue for chromophobe kidney cancer, we'll definitely work on that list. And we'll make sure that that list is in an accessible place, so anybody can see it at any time too. So, but we don't have those leads yet. We are definitely working on them.
For sure. And just we have two more before we wrap up. Is there any research specifically on hormone sensitivity of chromophobe?
Mm.
Um, this person's seen it mentioned in several articles. Would love to see the angle addressed. So anything that you can provide would be great.
We were just talking about that with Katie and others at the kidney cancer meeting in Denver because we were wondering if there might be some age-related and gender-related predispositions. And I mentioned that to Martin Voss, who has access to a large number of individuals with kidney cancer at Memorial Sloan Kettering, and he was going to look into it and see if he could confirm anything like that. And as it happens, we're very interested in hormonal biology as it relates to tuberous sclerosis. So just as I am thinking about this question, maybe we should be looking at that in the cell lines that we have and the components of this that we have available right now. Super interesting question.
Yeah, and I myself am particularly interested in it because I'm also a thyroid cancer survivor, and I know there are a few actually several kidney and thyroid cancer survivors where it's concurrent. So I'm not sure if the person is specifically talking about those type of hormones because we have so many in our body. But I think the hormone, personally, I think the hormone angle is a bit interesting. And our final question, unless Katie, you have any that you want to add on from the audience, is are you expecting any new treatments for metastatic chromophobe in the next year?
So are we expecting any new treatments? I think we're not going to have a brand new treatment proven to have efficacy in the next year because that would require a new clinical trial. But I do think we're going to learn more because I think we're going to learn more about who is benefiting from immune therapy. We know that some individuals with chromophobe are benefiting. So who are those individuals and how can we use this term personalizing therapy? How can we personalize therapy so that the people who would benefit are more likely to get that treatment? How can we predict in advance who's going to benefit? And again, this is something that Martin Voss and the group at Memorial Sloan Kettering seem to be making some progress on. So that's one angle. And then another angle is a clinical trial that's going on right now that combines immunotherapy with a tyrosine kinase inhibitor. And that will be interesting to see if we can learn something. That's not a trial only about chromophobe, but it might give us more information about who is benefiting in that trial. So I think we are making, not just me, but this village that has been created through many individuals, including COA, is moving toward helping to identify the best treatments that are currently existing, as well as moving toward brand new treatments for chromophobe kidney cancer.
I lied. We do actually have one more question if you don't mind staying on for an extra minute. But is there any research on existing meds that induce ferroptosis? I did not say that right, so forgive me and pretend I did.
Yeah, so we've thought about this a lot. So there are some drugs that are currently FDA approved and are believed to act in part by inducing ferroptosis. And I guess our thought is that probably these drugs are not potent enough inducers of ferroptosis to have clinical benefit in a kidney tumor like chromophobe kidney cancer. So even though we have studied some of these drugs in the lab, we don't think they're going to be the answer. We think we need a drug that is more specifically focused on the induction of ferroptosis. But we keep our eye on the literature, and maybe a drug that is already in FDA approved will turn out to be something that would have what we want to be a safe for the rest of the body, and to potently induce ferroptosis only in the kidney cancer cells. That's what we want, and maybe it is out there. We just don't know it right now.
And that's the amazing thing about research, right? I always say penicillin wasn't a thing until it was. And that's just the power of research. So hopefully we get that. Ooh. Well, thank you, Dr. Henske and Katie, for sharing your insights today. We've covered a lot of information and gotten a lot of valuable information. And I know this is incredibly just amazing for our community. So Katie, I'll turn it over to you to wrap up.
Yeah, so thank you everybody for joining, and Dr. Henske, thank you for giving us kind of a behind the scenes look of how research is done. I want to remind everybody that we also have our Giving Tuesday fundraiser running actually today for COA. This is actually the biggest fundraiser we've done in a single day, and we have a generous match donor up to $10,000. And we're just over $5,000 right now. So that goes through midnight. If anybody does want to donate, I'll just drop that into the chat if anybody wants a link for there. But most importantly, I just wanted to thank everybody for joining for the webinar and for participating and being a part of this village. So thank you, Dr. Henske, for joining us, and hope to see everybody on the next webinar as well too.